Welcome all. Myself Jayanti and I am going to discuss the topic computer aided drug design. Before entering into the topic, let me give the brief introduction. What is the necessity to know about the computer aided drug design techniques? These are the recently emerging techniques which are in addition to the in vitro and the in vivo methods which are used classically for the drug development process. These are the majorly used predictive methods for the drug design and development which can be very economical and can save lots of amount and time for the computational chemist and the research scientist. So, in order to develop a medicine, one should have a prior idea of the drug which have to be used. So, coming to the drug, it is a chemical substance that affects the process of the mind or the body, which is used in diagnosis, treatment, prevention of the disease or other abnormal condition. Coming to the process of design, it is nothing but an inventive process of finding new medications based on the knowledge of a biological target. Here, the biological target is nothing but a receptor or the protein molecule. The basic criteria followed by the drug molecule in order to design is it should be an organic small molecule, complementary in shape to the target, oppositely charged to the biomolecular target, that is the receptor molecule. In this diagrammatic representation, we can observe the interaction between the molecule of ligand and the receptor active site. So there must be an interaction with the target, bind to the target, and either activation or the inhibition function may take place. But we cannot solely say that the computer modeling techniques are only the basic for the drug design, but these are also frequently used for the better predictive methods. And coming to the drug design process, there are basically two types, that is based upon their process of design. They are classified into ligand based and the structure based. If the drug design process is happening through the structural information of the ligand which is going to bind to the active site of the receptor, it is said to be the ligand based, and based upon the structure of the molecule of receptor, that is structure based drug design. Coming to that ligand based drug design, it is relying upon the knowledge of other molecules that bind to the biological target of interest. It is used to derive a pharmacophore. Pharmacophore is nothing but a set of features which are collectively responsible for the therapeutic action exerted by a class of chemical compounds. Here we can observe this is a pharmacophore which can be generated by this design techniques. And this is the macromolecule which can be observed with the different amino acids like aspartate 25 and binded with the drug molecule. Coming to the structure-based drug design. Structure-based drug design relies on the knowledge of the three-dimensional structure of the biological target obtained through the methods such as X-ray crystallography, NMR spectroscopy, and the homology modeling. In this structure-based drug design, a receptor molecule is found out with various residues, that is hydrophobic pockets and hydrophilic residues. That is design drug to fit the pocket is the basic criteria in the back end of this design techniques. Using the structure of the biological target, drugs that are predicted to bind with to the target may be designed using various methods, that is interactive graphics, intuition of the medicinal chemist or the scientist, or the automated computational processes which are recently emerging. The various techniques used for the drug design are X-ray crystallography is one of them, and which is very important in describing a film with important coordinates that are liberated from crystallized molecule which is focused on the X-ray beam, and various coordinates are spotted on that film of which includes all the handling of data by the computer modeling systems, and this is used to determine the structural information about a molecule. Coming to the next method, that is NMR techniques. NMR is nothing but nuclear magnetic resonance. NMR uses much softer radiations and it is not applicable for the solid substances. It is used only for the liquid phase molecules. And three-dimensional information about the molecule can be easily obtained from this NMR techniques, and use it to examine small molecule macromolecule complexes such as enzyme inhibitor in the active site of the enzyme. And the last method for the structure-based drug design is homology modeling. Homology modeling is, in simple words, is nothing but building up of a 3D molecule structure, 3D structure of a protein molecule from the known amino acid sequences of that protein in reference to the standard protein molecule whose 3D molecule structure is already known. Here it is clearly given in the definitive manner, that is comparative modeling of proteins refers to the constructing an atomic resolution model of a target and an experimental 3D structure of a related homologous protein that is used as a template. Coming to the computer aided drug design. What is the necessity? Why these computer aided drug design techniques are having this much importance in these recent days? That is, this can be used for the further implications and the biomedical applications which can be observed here, like selectivity, affinity, metabolism and adverse reactions and environmental toxicity, which can be further observed from the drug used as a medication. So these computer aided drug discovery techniques are basically based upon these two design process, that is structure based and the ligand based design. Coming to the brief introduction, it is represented as a computational method and the resources that are used to facilitate the design and discovery of the new therapeutic solutions. So by using these computer aided drug design techniques, a new molecule can be identified with more computational source and various sources of literature which are used for the drug design and development. And coming to the history of the drug development, what is the history? Screening for the new drugs. What is the necessity for the screening of new drugs, even if there are many drugs available in the market nowadays? Coming to the screening, uh, the major sources of the screening of the drugs are plant sources and the accidental observations. There are various plant sources or the natural products which are responsible for the medication activity. That is example, we can consider foxglove for the treatment of heart failure, and there are many accidental observations for the drug molecule identification. That is example, penicillin, which is identified by Fleming from the effect of mold. And the modifications for the improvement, there are many modifications done for the improvement of the existing drugs which can enhance their activity and further act in a very effective manner. So these modified compounds can be engineered directly by the modification of the genes. Mechanism based drug design. This is nothing but when the disease process is understood at the molecular level and the target molecules are defined, drugs can be designed specifically to interact with the target molecule in such a way as to disrupt the disease. So, based upon the mechanism based drug design, if the active site of the receptor molecule is identified and the target is designed in such a way that ligand molecule acts directly on the target, and the further mechanism of action can be disrupted and the disease can be healed. The basic mechanism of computer aided drug design process is here. We can observe the various steps involved. The first and the foremost step is to identify the disease, the causative disease for the person, and the target, what is responsible for the causing of disease. After identification of the particular target, the protein sequence responsible for the target active site is identified. And then coming to the structural determination, the protein sequence structure is obtained from the various sources like chemical structure database and CSD, that is, and protein data bank, that is PDB. The 3D structure can be obtained and its known actives and the structure can be determined. Further, the lead search takes place and the lead structure can be obtained, which can be optimized, that is, the lead optimization occurs. And further lead optimization carries towards the drug candidate development and further clinical trials, that is phase 1, phase 2, phase 3, so on. The clinical trials takes place and then finally the drug marketing can be observed. This whole process can be done by the help of chemoinformatics, bioinformatics, biologists and the medicinal chemists, which has major complicated part involved in the every step of design process of a drug. Coming to the first and the foremost step, that is selection of the disease. The first step in the design of the drugs is to treat the diseases and to determine the biochemical basis of the disease process, that is to identify the specific receptor that the macromolecule responsible for the disease, and the normal functioning of the disease pathway has to be identified, and the receptor molecule active site have to be identified, that is the target selection. From this target selection, the potentially many ways in which biochemical pathways could become abnormal and result in the disease. So the molecular basis for the disease can be selected, that is the target can be identified to disrupt the process. And coming to the target selection, there may be three main targets, that is enzymes, receptors and the nucleic acids. The structure determination. Once we identify the target, the structure can be determined from the various sources, that is PDB, CSD. Coming to the PDB, this is an open source for obtaining the structure, 3D structure of a molecule. For example, if the PDB ID is given, the structure can be easily downloaded and the crystal structure of that whole molecule can be studied for the active sites, and the drug design technique can be easily carried out. And this is the open interface of the PDB homepage. And determination of the active site of the target protein. How the active site can be identified. Only a small part of the lead compounds may be involved in the appropriate interaction. So it must be identified for the target selection. And for the activity of the ligand have to be focused on this active site, which is very responsible for the disruption of the disease pathway. The relevant groups on a molecule that interact with the receptor and are responsible for the activity are collectively known as pharmacophore, as we already discussed. Selection of the ligands of the drugs. This is the very important and the essential step which is handled very carefully. That is, it has a very important role, that is, because ligand structure is very specific for each and every molecule. That is, receptor molecule based upon its structure, a ligand is gets into fit into the particular specific receptor. So this selection process is a very crucial step in the drug design and development. This can be followed by the important computer aided drug design technique, that is molecular docking. Docking, in simple terms, it is nothing but and maneuvering of drug molecule into the cavity of the protein. Docking is a whole process of orientation of one molecule to a second when bound to each other to form a stable complex. Here we can observe this molecular recognition site of the protein molecule and the ligand getting and binding into that specific receptor site. Here comes various softwares used for the docking, that is dock, autodock, MOE, that is molecular operating environment, enable user to predict favorable biological target ligand complex structures with reasonable accuracy and speed. The backend algorithm running for docking is of various types, and the major thing is induced fit, while flex is capable for it. And the various softwares uses various algorithms, that is doc uses incremental construction algorithm, and gold software uses genetic algorithm, and glide uses Monte Carlo search and the genetic algorithm search. The majorly used software for the docking purpose is Autodock, which is a suite of automated docking tools. It is designed to predict how small molecules, that is substrates of the drug candidates, get and bind into the receptor whose 3D structure is already known. This is the representation of how the 3D molecule of the receptor and the drug molecules get binded. And for the visualization of this whole docking complex, VMD, Visual Molecular Dynamics, is used. This is the interface for the VMD, and these are the various icons and the graphical user interface used for the visualization. And retrieving the 3D structures. How to retrieve these 3D structures is the basic phenomenon lying under this visualization. The first step for the protein visualization is to extract the protein structure from a protein structure database, that is PDB or the CN3D format, which would serve as input for the 3D visualization programs. And this is the basic link we can proceed for the retrieving of 3D structure from the PDB database. And this is the home page which we can have as the interface for the PDB. What is VMD? VMD is nothing but Visual Molecular Dynamics is a molecular visualization program for displaying, animating and analyzing large biomolecular systems such as proteins, nucleic acids, lipids, etc. using 3D graphics and built-in scripting. VMD supports various computer running programs, that is Mac, Unix, Windows, and is distributed free of charge and includes source code. It may be used to view more general molecules, as VMD can read standard protein databank files and display the contained structure. The major use of this VMD is that it provides a wide variety of methods for rendering and coloring a molecule. It is not only used to animate and analyze, it is also used for the trajectory of the MD simulations and also acts as a graphical front end for an external MD program by displaying and animating a molecule undergoing simulation on a remote computer. What are the key features which are used for the VMD? General 3D molecular visualization with extensive drawing and coloring methods. It is also an extensive atom selection syntax for choosing subsets of atoms for display. The visualization of dynamic molecular data can be used by rendering high-resolution publication quality molecule images. Building and preparing systems for molecular dynamic simulations. Interactive molecular dynamic simulations with the extensible source code written in C and C++. Not only these, many other key features of the VMD are its movie making capability and molecular analysis commands, and there is no limitation as per the number of molecule and the trajectory frames are concerned, except the available memory is only a little bit problem in VMD. It almost supports all the molecular data file formats, and a visualization of the volume data is also facilitated in this VMD. So coming to the final benefits of the CADD, what we have discussed. Drug discovery. It is the major process for the drug development and the output of a drug as a medication. So this CADD is a major source for the drug discovery process. Elimination of the compounds with the undesirable properties. So this acts as a major source for the elimination of the drug with these undesirable properties. That is, design of in silico filters to eliminate the compounds with such properties and select the most promising candidates that are suitable for the drug processing. It also helps in the identifying and optimizing new drugs. That is, chemical and the biological information about the ligands or the targets to identify and optimize new drugs can be processed by these computer-aided drug design techniques. And the most useful and the utmost advantages are time-saving process. The process of drug discovery and development is a long and difficult one, and the costs of developing are increasingly rapid. Today, it takes approximately 10 years and $100 million to bring a new drug into a market. So, this is the best method for the decreasing this cost and the time limit for drug discovery. Coming to the reduced cost, the use of the new computer-based drug design techniques has the ability to accomplish both of these goals and to improve the efficiency of the process as well, thus reducing costs. And also, it improves the quality of life. The emphasis now is not just on finding new ways to treat the human disease, but also on improving the quality of life of people in general.